Thanks very much, everybody, and appreciate the opportunity to share the Invion story with you. If I can get these slides to work. So we have a novel next-generation photodynamic therapy, which is a combination of light and drug. And we're targeting primarily in multiple cancers, and we're also doing some work in infectious diseases as well. I'll let you read the disclaimer. But essentially, we are at clinical stage. So we actually cured cancer in mice, but people don't really care about that so much. So we're actually at clinical stage right now. And we have built a portfolio of over a dozen patent families, which we filed over the last couple of years. And we'll be sharing with you our clinical program and some clinical trial results, including the results of our phase 2 trial that was done. We actually announced yesterday, and I'll share some of the results of that with you today. We're also have filed a CTN for a non-melanoma skin cancer phase 1 and 2 trial, which we're starting recruitment over the coming month. So there are a number of catalysts coming up for the company. And as mentioned, our primary focus is on oncology, so multiple cancers, and we're also doing some work in infectious disease. And as a small biotech, we also want to make sure we partner as part of our core sort of our values. So we're partnering on the research side with the Peter MacCallum Cancer Center, as well as the Hudson Institute of Medical Research, and we're building a further portfolio of other partners. On the sort of portfolio side and development side, we're partnered with a couple of Korean companies. One is a pharma company called Hunlim Pharm, and the other is Dr. INB, and I'll talk about those in a little bit. So what's our technology? Technology essentially is a photosensitizer or a drug, which can be administered topically or IV or systemically. And the drug's designed to selectively accumulate just in cancer cells and not healthy cells. So by itself, nothing happens until you shine a frequency of light, and that generates reactive oxygen species. So that oxidative stress, because it only localizes in cancer tissues, is cytotoxic and just kills the cancer cells. That's the first mechanism of action. The second mechanism is essentially activating the immune response, and I'll talk about some of the work we've done in that space. As mentioned, our primary focus is in cancer, and I'll spend most of today's talk in that area. We've already got results from the prostate cancer trial, phase 2 trial, investigator-led trial I'll go through. We're in the process of recruiting for non-melanoma skin cancer, and we're working on an anogenital cancer trial with Peter Mac, which hopefully in the coming year or so we can start kicking off. And we're looking at a couple of other areas as well. And if we have time, I'll talk about some of the infectious disease work we're doing. Now, we've been very unloved and very much below the radar screen, and there's a good reason for that, and I don't blame the market for it. It's really the background to where we're at. So we were actually meant to start clinical trials several years ago using an older version of our photosensitizer. And this little thing called COVID came in the way, and we actually had to cancel our trial plans. And we used the opportunity to essentially go back to discovery mode and actually develop literally hundreds of novel new compounds. And we picked one of them, INV043, to be our lead cancer candidate. And that's what we've spent the last couple of years working on the preclinical work and the CMC, or the chemistry manufacturing control work, so that we'd be ready to do a clinical trial right now. And so during this last several years, it's just been very uninteresting for the market because we've had mice cancer cures, we've had things that the market doesn't care about. We are at the stage now where there are things that we hope that the market really cares about. Safety in humans, efficacy in humans. And so I think you'll be seeing a number of catalysts over the next 1 to 2 years that hopefully will be groundbreaking. So let's talk a little bit about our lead candidate for cancer, INV043. We've actually tested it across a number of cancer models, animal models, pancreatic cancer, T-cell lymphoma, triple-negative breast cancer, and we've found regression ability in all those cancers. So we know it works across multiple cancers. We also know it's far more potent than other photosensitizers and PDTs out there. It's a technology that's been around for a while, but the old existing technologies have a lot of off-target issues. So we're 600 times more potent than teleporphin sodium, which is which is an approved PDT. But just as importantly, our safety profile's really good. We've actually tested at 100 times the therapeutic dose IV and haven't found any toxicity issues. So it's safe and it works across a number of cancers. It also stimulates the immune response, and I'll talk about some of the studies around that a little bit later. So let's start with the most recent findings, which is essentially our phase 2 prostate cancer trial. It was actually an investigator-led trial. Originally, it was meant to be a 75-person study. And, our friend COVID actually impacted there. So we eventually only recruited 41 patients. And of those 41, 25 happened to be recruited during COVID, which meant that we couldn't get all the measurements and markers that we actually wanted. 16 of the patients actually were recruited after COVID, and they're the ones which we've extracted the data for, which I'll share with you in a little bit. Our primary endpoint was efficacy, which was using the RESIST framework, which essentially measures change in tumor size. And we also had a number of other secondary endpoints around safety as well as efficacy. The treatment protocol was essentially administering our PDT over 6 times during a 9-week period for every patient. And it was administered sublingually, so under the tongue, which means that it actually gets into the bloodstream. And so we have systemic safety data around that as well. So what do we find? Firstly, it's very safe. So we actually did not find any serious adverse events. And actually, we tested each patient of the 16 patients 6 times for questionnaires. And you see the results at the bottom. Very mild type of results. We had some nausea, fatigue, fever, a little bit of soreness. That's pretty much it, which is very important because if you think about, for the gentleman in this room, the side effects of conventional prostate treatment cancer can be pretty devastating. Incontinence, ED, infertility, et cetera, et cetera. So it's a potential here to have a very safe alternative to prostate cancer treatment apart from just monitoring. Let's look at efficacy. So I mentioned the primary endpoint was around the RESIST or measuring the impact on our tumor size. And what was found was about 40% of the patients in this group actually had a positive response after 3 months. So, and 10% of the patient actually had a complete response. So given the safety profile, that's pretty interesting. We also had another marker called the PSMA PET test, which essentially tests for a prostate membrane-specific antigen. And basically all 16 patients at the start were positive for it. Only 9 were positive at the end of 3 months. So there was a 44% response rate. So this is a monotherapy trial. So that actually gives some pretty promising results that would justify some additional larger studies, particularly given the safety results. If we take a step back here, we've already got some human data. What are the longer-term pipeline opportunities? So one of the interesting characteristics of this technology is the ability to have both diagnostic and therapeutic characteristics. If you look at, there's 2 wavelengths of light that activate this compound. The first is actually in the visible red light spectrum, and that generates the reactive oxygen species that kills those cancer cells it localizes in. But if you flick the switch into the violet spectrum, it actually just fluoresces. And that's actually what you're seeing on the left-hand side. So those pink spots there, and the pink is essentially the cancer cells, which are being fluoresced. And we've literally used a $20 light that you can buy on the internet, and we've used an iPhone, and you see that image. So bearing in mind that about 30% of tissue that's cut out by surgeons is healthy, and 30% of tissue is typically missed, it could become a very good diagnostic tool in addition to the therapeutic sort of potential here. The selective targeting, as you can even see with the little metastatic pink spots, actually shows that it's got very good precision targeting for the cancers. And so that addresses the ablation and the regression of the tumors. The second side of the equation is on the right-hand side, which is essentially the immune response, or the activation of the immune response. This was a pilot study done by the Hudson Institute, and basically it was performed using a triple-negative breast cancer model on an immunocompetent mouse. So triple negative is typically chemo-resistant, pretty nasty, typically metastasizes. And if you look at the bottom two pictures, you can actually see that this mouse basically started with a primary lesion that totally disappeared. There was no scarring, no sign of metastases, and by itself, that was very interesting. The second interesting element was a week later, we tried to reimplant the tumor and it would not grow. And based on the immunological work we did afterwards, we actually found that it activated the T-cell immune response. So obviously, if we can translate this into the human context, now we're talking about reducing recurrence, potentially addressing metastases, potentially having abscopal effect. So that's the holy grail stuff. We're not banking on it, but there's a possibility. In terms of the immune response as well, we've done some work in combining it with existing therapies. We've done a couple of studies with both Peter MacCallum Cancer Center as well as the Hudson on combining our therapy with checkpoint inhibitors. As you know, checkpoint inhibitors are part of the immunotherapy suite of treatments. It's modalities for cancer. It's actually, when they work, they actually work really well. The problem is they normally don't work. It's about a 12% response rate. In fact, that's actually what Peter Mac found in the standalone checkpoint inhibitor group in this study. We increased that in combination to 80% response rate. So if we can get anywhere close to that, that changes quite dramatically the potential patient outcomes, particularly given that a number of these blockbuster drugs are actually coming off patent in the next few years. So here's an opportunity to extend patent life in combination. So basically two tumor types, triple negative versus anal squamous cell carcinoma, 2 institutes, 2 routes of administration, consistent results. So we're very excited about bringing this forward into clinical application. When you talk to clinicians, we're trying to make it as easy as possible to fit into their protocol and their regime. So rather than them fitting into our square peg round hole, we're trying to fit into a clinician's sort of patient needs. So the first question they often ask is, how do you get the drug there? Well, we've got a topical formulation and we've got an IV formulation. And the same IV formulation can be administered intratumorally or sublingually, as we did in the prostate trial. So getting the drug there is pretty straightforward. Now, how do we get the light there? Most people think of topical administration with a laser light or an LED. Now, as long as we can get a fiber optic cable in there, we can actually get light through an endoscope, just a little fiber optic cable through an endoscope, keyhole surgery, obviously open surgery. So there's a wide range of indications that can be targeted through this. And that brings us forward to our pipeline of trials. We've completed the prostate cancer trial, which is phase 2 trial. We've found very good safety data and promising efficacy signals, and would like to see and explore how we can take that forward. We've got CTN and human ethics approval to move forward with our non-melanoma skin cancer trial, and that's using a topical route of administration. That trial is actually, we're looking to start recruiting ideally in the coming month or so, and it's designed as an adaptive trial. So it starts off with the first arm around safety, and the second and third arm is around dose optimization and efficacy. And so we're pretty excited about the potential there. And because we're looking to use the same topical formulation into our anogenital cancer trial, we are ideally looking to leverage the safety data from this trial into potentially a phase 2 trial with an anogenital cancer with Peter Mac. I won't talk too much about glioblastoma. It's preclinical work. The main point here is that program is being funded by Hunlim Pharma. So that sort of takes the cash flow load a little bit off. But let's talk a little bit about our upcoming trial on skin cancer. Why do we focus on skin cancer? Well, firstly, it's relatively cost effective in this market. You've got to get as much bang for the buck. And because it's topical, the non-melanoma skin cancers are superficial cancers. There's really no systemic implication, very safe. Clinicians are comfortable with it. And so it's easy and cheap to basically administer this. And because of that, there's also a faster path to registration to market. As I mentioned, there's also synergies because we can use the safety data and the same formulation into, for example, an anogenital cancer trial, which we plan to do. And it is one of the most prevalent cancers in the world. The other aspect is there's a pretty big unmet medical need, including the fact that originally I was surprised when I saw the data is more people die from non-melanoma skin cancers than melanoma. The incidence of melanoma is obviously higher, but because there's such a high rate of non-melanoma skin cancers, it becomes pretty important. And in fact, every 2/3 of Australians by the age of 70 will actually have been diagnosed with non-melanoma skin cancer. So it's a pretty big unmet need. Now, obviously, we have to see where we play in this space. And if you look at current standard of care, most standard of care are essentially surgical interventions. So you can see that on the bottom right-hand corner of the chart. It works pretty well. The efficacy generally is pretty good. The downside is the cosmesis. There's some pretty nasty scarring that occurs. And there's actually an approved oral PDT called Metvix that's owned by Galderma that helps address the cosmetic issues, but the efficacy is not as good as surgery. What we hope we can achieve is, and by the way, the other downside of Metvix is it's very painful. About 10% of patients can't even finish the treatment because it's too painful. So what we're trying to do, we have something that's 600 times more potent than another photosensitizer out there. We haven't noticed pain in our preclinical work. We have the same mechanism and actually, frankly, more precision. So we're anticipating equivalent efficacy. We don't need superior efficacy. We just need equivalence. And also having similar cosmetic outcomes without the pain. So long story short, go to a patient. Would you like to treat your cancer with or without scarring? And if we can achieve that without pain, we have a potential to displace the market. That's where we're looking at in terms of non-melanoma skin cancers. The other area about our modality is really creating impact. And, we're also living the economic realities of trying to get a drug approved and actually getting revenues from it, et cetera, et cetera. But when you take a step back, the cost of new drugs is really quite tremendous now. If you look at the new FDA drugs approved last year, in 1 year, the price went up by 35%. And that's in the US, which is one of the richest countries in the world. So if you think of all these new drugs that are coming out, they're becoming less and less affordable to the masses and frankly to the rest of the world as well. The way we look at it, because our technology localizes on multiple cancers, irrespective of patient, we have a precision medicine that actually doesn't require personalization for every patient. That creates basically economies of scale. We're also a small molecule-based compound. So again, that can be scaled up. And our solution and our light is actually a very simple outcome. So we believe we can have a solution for patients that's going to be a smaller amount than the $300,000 US dollars per treatment or per patient costs that are really coming into play in the new drugs out of the US. Similarly, if you think about our neck of the woods, 60% of the world is in Asia. 6% of patients in FDA drugs are Asian. There's really a lack of focus on unmet needs and cancer for the majority of the world. That's the reason Stanford Medical School started a Center for Asian Health Research. And I joined their board quite recently. and we're collectively trying to see how we can make a difference in addressing these unmet needs for the masses in the world. Okay, I don't know how our time is. Wind up?
Take a few questions.
Okay, well, what I might do now is just
the best part of the presentation, by the way.
Oh, really? Okay, all right. I'll just quickly mention we are touching on infectious disease. It's not our primary focus because we have to target, but what we're trying to do is essentially find opportunities to address the antimicrobial resistant issue, which is one of the top 10 threats to humanity. We've tested a number of our compounds, which are broad spectrum antimicrobial, bacteria, viruses, and fungi, and found it to be very sensitive and very effective. But what we don't want to do is put a lot of resources into that. So our solution has been to find someone else that'll fund the program, which is what we've done with HPV, which is a big precursor to anogenital cancers. It's a Korean company that's funding ourselves to proof of concept clinical trials in Korea. And we're also looking at periodontal disease because that's an area where about a half of Americans over age 30 have some sort of periodontal disease. And with an aging population, if you have a implant, you're about a quarter to a half of those patients actually get some sort of peri-implantitis or infection / inflammation. We believe you get a little mouthwash, wash it every day, put a light in, reduce the microbial load, let your gums heal. We believe we have potential to actually substantially reduce that incidence of peri-implantitis. So that's pretty much it. And maybe I'll open it up to questions.